The males on the high-energy control feed showed a huge spike in VGL3 expression. It was 1.58 times higher than the low-fat group.
And the low-fat group.
No significant change over time. It stayed flat.
So the high-fat diet didn't just make the fish fatter, it actually unlocked the transcriptional activity of the VGL3 gene.
It's a direct gene-environment interaction. The lipid intake is signaling the genome, basically saying, okay, we have the energy reserves, fire up the maturation machinery.
This is a powerful idea for nutritionists. We can feed the genome.
It opens the door for precision nutrition.
A whole new era of communication in the global aquaculture industry is coming. Now you have the brightest minds in aquaculture right in your pocket. And what's best? You can listen to all of them while driving to a farm, traveling, or running errands. It's never been this good, and it's never been this simple. Welcome to the Aquaculture Podcast Show, the first AI-based podcast in aquaculture, where you'll find cutting-edge insights in everything that's working in aquaculture, nutrition, health, and production. Inspired by the 2024 article titled Sex Development and Nutrition-Dependent Expression of a Major Age at Maturity Gene in Atlantic Salmon by Åsheim, Eirik Ryvoll et al.
Welcome back to the Deep Dive. Today, we're getting right into production efficiency, specifically in Atlantic salmon. There's one word that I think gives every production manager a headache. Grilsing.
Oh, absolutely. It's the biological bottleneck. You have a fish growing perfectly on target, and then bam, it pivots to mature early.
And your profitability just tanks. You're suddenly dealing with flesh quality issues, skeletal problems, a harvest you never planned for.
Exactly. For a decade, we've been focused on the genetic driver, the VGL3 locus. Everyone knows it as the age at maturity gene.
Right. If you're in broodstock management, VGL3 is on your dashboard. We screen for it. We select for the late alleles, but we've basically been treating it like a light switch.
On or off, early or late. That's been the wisdom. We focus so much on the genotype, the blueprint, but not the transcriptomic.
We never really understood how that blueprint was being read in real time.
Yeah, we've just assumed the late gene maturation was suppressed, period.
But a new 2024 study by Åsheim, Ryvoll, and their team is showing us it's not that simple at all.
No, it's not. The paper's titled Sex, Development, and Nutrition-Dependent Expression of a Major Age at Maturity Gene in Atlantic Salmon. And it just completely dismantles that light switch theory.
It really opens up the black box, doesn't it? They've shown that VGL3 isn't just a brake. It's more like a complex throttle.
A throttle that responds differently depending on the fish's age, its sex, and critically for our listeners, what you're feeding it.
And before we get into the paradox, the quality of this data is just fantastic. It's not a snapshot study.
No, it's a longitudinal study, the gold standard. They tracked gonadal VGL3 expression from the pre-pubertal stage all the way through multiple spawning seasons. Some fish up to 4 years old.
That gives you the full motion picture.
Right. And they used RT-dPCR, which gives you absolute quantification. It's not just more or less expression, it's counting the actual RNA copies.
Like putting the fish on a calibrated scale instead of just guessing its weight.
Precisely.
Feed formulation software is stuck in the past. Most tools haven't evolved beyond basic least cost optimization. A Diet Forge changes that. It's an AI-powered platform with nonlinear ingredient modeling for enzymes and additives, cost per gain and maximum profit optimization, and real-time collaboration like Google Docs for nutrition. Every recommendation is grounded in world-class research, built for modern nutritionists who need more than legacy desktop software can deliver. Work seamlessly with your team and optimize the way modern operations demand. Try dietforge.ai free today. Feed formulation, finally modernized.
So that brings us to the expression paradox. The dogma has always been that VGL3 is an inhibitor. It stops the HPG axis from firing.
The idea being it suppresses Sertoli cell proliferation in the testes via the hippo pathway, keeping the fish immature.
Correct. And to be fair, the data from the first spawning season actually confirmed that.
It did. The immature fish had way higher VGL3 expression, about 1.6 times higher in ovaries and double in testes.
So in season 1, the rule holds. High VGL3 means stop. It keeps energy focused on somatic growth.
But then they kept watching. And in the lead up to the second spawning season, everything flipped.
It completely inverted. From February to August, as the fish were maturing for the second time, the expression of VGL3 didn't drop. It actually went up.
It increased. So high expression stops puberty in year 1, but accompanies it in year 2. That just contradicts everything we thought.
In the testes, it went up about 1.2-fold per month. If it were just an off switch, that just shouldn't happen.
So what's the logic? Why would it repress one year and promote the next?
The authors propose a checkpoint hypothesis, which I think is very convincing. They suggest the downregulation, the release of the brake, is specific only to the early stages of spermatogenesis.
Okay, so it's a window. Once you're past that initial cell proliferation checkpoint.
Exactly. Once the cellular machinery is built, the gene's role seems to change. The later stages of maturation seem to involve a gradual upregulation. It's not just blocking the door anymore.
Which is a crucial detail for anyone developing biomarkers, right? High VGL3 in year 2 doesn't mean immature. Context is everything.
And context brings us to the biggest split in the data.
The difference was just stark. They looked at the gonodosomatic index, the GSI, against gene expression in that August run-up to the second spawn.
A complete mirror image.
For the males.
A strong positive correlation. Pearson's R was 0.79. As testes got bigger, VGL3 expression went up right alongside.
And for the females.
A strong negative correlation, R of negative 0.92. As the ovaries gained mass, VGL3 just plummeted. It's incredible. Same species, same gene, but opposite regulation. It has to be an interaction with the hormonal landscape.
Specifically, estrogen signaling.
Right. In males, you need some estrogen early for spermogonial stem cell renewal. But in females, the massive estrogen spike comes later for vitellogenesis, for building the yolk.
So the theory is that VGL3 negatively regulates that estrogen signaling.
That seems to be it. For a female to get that massive ovarian growth, she has to turn off VGL3 to let estrogen run wild.
But the males are past that early phase, so they can tolerate and maybe even use high VGL3 during that later growth.
This is a huge deal for breeding programs. You need sex-specific selection indices. You can't just treat the broodstock as one group.
Okay, let's pivot to the feed factor. This is where I think the immediate ROI is for everyone in production.
For sure. We all know fat fish mature early. But this study asks, is it just growth, or are the nutrients actually flipping genetic switches?
They set up a control feed, your standard 17 to 26% fat diet, versus a low-fat diet around 12 or 13%.
And what they found was fascinating. In Season 1, diet made no difference to expression. But leading up to Season 2...
That paradox phase.
Exactly. The males on the high-energy control feed showed a huge spike in VGL3 expression. It was 1.58 times higher than the low-fat group.
And the low-fat group.
No significant change over time. It stayed flat.
So the high-fat diet didn't just make the fish fatter, it actually unlocked the transcriptional activity of the VGL3 gene.
It's a direct gene-environment interaction. The lipid intake is signaling the genome, basically saying, okay, we have the energy reserves, fire up the maturation machinery.
This is a powerful idea for nutritionists. We can feed the genome.
It opens the door for precision nutrition. If you have a batch that is genetically predisposed to grilsing, maybe you can deploy a maturation dampening feed with a lower lipid profile during that critical window.
You're manipulating the metabolic signal to delay the gene's response.
Exactly. It's proactive biological management, not reactive culling.
And just to round it out, they looked at some other markers too. It's not just VGL3 working in a vacuum.
No, they tracked AM, anti-Müllerian hormone, which is a known spermatogenesis inhibitor. It correlated positively with VGL3 when the testes were undeveloped.
Like 2 guards on the door.
Right. But then as maturation kicked in, AM dropped off, 1 guard left his post. It's a whole network.
So let's synthesize this. What's the actionable takeaway for a production director?
The takeaway is that this gene's expression is plastic. It's not destiny. It can be managed.
And that means we have levers to pull.
Precisely. We have the genetic lever, but we have to use it with sex-specific data. And we have the environmental lever through precision feeding. The genetics team and the feed team have to be in lockstep.
You tailor the diet to the genotype to optimize growth while keeping that maturation dial turned down.
That's the goal.
There's one final thought I want to leave everyone with. This gene, VGL3, it's conserved across vertebrates. We have it.
It's an ancient, fundamental regulator of puberty.
But seeing it function in opposite ways between male and female salmon, it raises this question about evolutionary trade-offs. Why couple growth and reproduction with the same lever, but pulled in opposite directions?
And for us, the challenge is, can we decouple them?
Yeah.
Can we use this molecular knowledge to get that massive somatic growth without triggering the reproductive penalty?
That's the holy grail, isn't it? Decoupling growth from maturation.
It is. And studies like this mapping the transcriptomic landscape are how we'll get there. We're moving from farming fish to engineering biology.
It's fascinating work from Åsheim and the team. That's all for this deep dive. Keep your feed precise, keep your stock healthy, and we'll catch you on the next one.
If you enjoyed this episode, make sure to follow the Aquaculture Podcast Show on your favorite platform. And don't forget to follow us on LinkedIn, Instagram, and Facebook to stay updated on the latest episodes and industry insights. Thanks for tuning in, and we'll catch you next time.